With this, I would like to start with our introduction for the next speaker, Maxim Cazorla. He co-leads a research team and serves as a scientific director of the Neurotech Center at the Institut de Neurosciences de la Timone, Aix-Marseille University. In 2023, he co-founded the company Ateca TX, a pharmaceutical startup dedicated to developing innovative therapies for neurodegenerative diseases. He earned his PhD in neuroscience from Paris-Sorbonne University and conducted several postdocs at Columbia University and Institut Curie. And currently he's focusing on brain rewiring capabilities and the development of alternative models for studying neurodegenerative diseases. So the stage is yours, Maxime. Thank you for being here.
Thank you for the invitation. Thank you for the nice introduction. And so I changed a bit the title today to focus more on what we've been working on lately in the lab, where we're trying actually to redefine brain disease modeling and drug testing within the context of the 3 Rs, and using mostly this, what you can see here on this slide, this what we call organ chips, even though I don't really like this term, but you will see why afterwards. So why modeling brain diseases? So as you know, we are currently using 3 different kinds of studies, let's say 3 different level of approaches. The first one being probably the most important one, the human studies, using functional imaging, EEGs, mostly used also for genetic studies to observe and determine symptoms associated with specific mutations, for instance, and diseases. We are extensively using animal studies because they are very convenient. They allow for genetic manipulations, intoxications. We can study their behavior, circuits, functions, etc. So they're extremely useful. And the last, probably the most simple approach is probably the in vitro studies, because they are very simple, very reproducible, and they allow causative results by studying the mechanisms and allowing large, or at least drug screening, not necessarily very large, but you can do drug screening more easily with in vitro studies. But all of these approaches have limitations. And of course, for the human studies, the main limitations is that you observe symptoms, and providing indirect results, you can correlate with a symptom, but it's not necessarily the cause. And of course, you have multiple confounding factors that are very difficult to extract. For animal studies, people complain most of the time that they are not always relevant for the human, as we have heard before in the presentations, and also because we are facing an incredible urge to reduce animal research. And for in vitro studies, they are very nice, very simple models, very useful model, but they are extremely limited. They are not necessarily very relevant for the complex organization and the function of the brain, which is probably one of the most complex organ in the human or in animal kingdom. So there is a need for better brain models. And actually, we've been looking for a model that would bridge the gap between all of these approaches. So it's very ambitious, of course, as a question, but the idea was to have a model that would keep the relevance of human studies to be relevant for the human brains. And for instance, by keeping the genetic landscapes and epigenetic landscape of the human brain, it has to be as useful as animal studies to allow manipulations and provide causative results, and keep the ease of access of in vitro studies and allow for mechanistic studies. And we believe that in the last decades, progress in microfabrication and stem cells, and particularly human stem cells, have provided a new way of modeling human neural networks, and that being on a chip. So if you combine stem cells with microfabrication, then you can produce this, what we call brain on a chip. Once again, I don't really like this term. And once again, I will discuss about that. But they are very convenient because you have access to human iPSCs, human-derived cells, then you are relevant for the human brain. It's capable of reproducing the complex architecture, at least a part of this architecture of neural networks, because of the microfabrication, and also because it's in vitro, then it becomes compatible with mechanistic studies. So in practice, how does that work? As you see, it's very simple to use. You have an idea of circuit of interest, a DC2 model. So you just draw on your favorite paper or blackboard, you draw a design that will resemble the constraints of your circuits of interest. Then if you have a nice collaboration, as I have with Benoît Charlot from Montpellier University, you can do the microfabrication using a lithophotography here. And then you produce them. There are small pieces of silicon, a biocompatible material, which is in which cells are happy to live with, and very easy to replicate. And then you can use them again on different kind of assays. Here it's video microscopy coupled to microfluidics perfusions, for instance. To design your chips, it's very easy. Once again, it all depends on the circuits of interest. So you can mimic different types of architectures depending on the network. So you see here a few examples of them. So all of them have different designs that are instructed by the network of interest. In that case here, we were interested in the corticostriatal pathway circuits. And for that, we were just using cortical progenitors seated in the presynaptic compartment, the striatal progenitor seated in the postsynaptic compartment, and then using different sensors, lentivirus infection of these chambers. They are based on they are fluidically isolated because they are based on microfluidic devices. So it's very easy to infect only one specific chamber with your favorite marker or sensor, and then you can perfuse here your compound of interest that you want to test. In this case, it will be perfused in this very specific chamber, and then you can look at different readouts. In these cases here, very briefly, we are capable of isolating axons, cortical axons connecting striatal cells, so we can follow the trafficking, in this case, trafficking of mitochondria. We can look at glutamate transmission using a gluesniffer sensor, for instance, specifically here in the synaptic chamber where cortical axons connect striatal dendrites. And you can look at the density of these synaptic contacts, the population activity of the striatum in response to cortex, etc. So this is a very convenient system. Those are just a few examples that we've been using lately in the lab, but you can use pretty much everything that is compatible with cell cultures. We have also connected these microfluidic chambers to MEAs, microelectrode arrays. So we've heard about multi-channel system before. So this is the system we were using here. Now we have implemented the 256 electrode microelectrode array. And now we're able to record or stimulate every chamber, so either the cortex or the striatum, in presence of a compound of interest that will, here you see, completely obliterate the communication between the cortex and the striatum. Once again, done in collaboration with Benoît here. So now, as I said, we have access now to human cells, and thanks to another collaboration with Emmanuel Livet here at Aix-Marseille University, we are now transforming this rodent chip, corticostriatal network chip using human cells, and we are making a human corticostriatal a chip right now to study, and so we will compare the different results that we got on the readouts I showed previously to see if what we have with human cells is comparable to what we got with rodent cells, and it will give us a good readout of how we can compare results and how you will see a drug of interest would have some clinical efficacy in human cells. So this is an ongoing work. I will come back very briefly at the end here. But what I wanted to do first is to show you examples of how we use these chips, these very little pieces, like very easy to use microchips to design new models, new brain disease models. So in this case, the first one we tried was Huntington's disease. And at that time, there was a big question. I think it's still a big question in the field to know whether Huntington's disease is a cortex or striatum disease, because as you probably know, the Huntington's disease is traditionally viewed as striatal disease, because this is where you have like the most degeneration. But some people tend to say that probably cortex is the first one to degenerate in the process early in the disease, and that will drive the degeneration of the striatum. So we modeled this disease using a cortical and striatal progenitors from different Huntington's disease mouse models. And we reproduced the chips, and then we looked at different markers. I'm just showing a few of them here. And one of the most striking was probably this calcium imaging of striatal cells in response to spontaneous activity of the cortex. And you can already see that in Huntington's disease chips, you have this very prototypic response of the cortical of the striatal activity with huge burst of activity, highly synchronized, and this is interspaced with long period of silence. So it was a very striking phenotype that was easy to screen. So then because of the versatility of this chip, we said, oh, instead of just looking at full wild type or full Huntington's disease network, now let's make hybrid networks where you have a healthy cortex connecting a diseased striatum and the other way around. And then we reproduce the same kind of experiments, and we see the different dysfunctions on the different readouts. And long story short, what we found is that actually, as long as the cortex is healthy, then the whole network will be healthy, will behave as a healthy network. So meaning that if you're looking at something, particularly a readout of the striatum, even though it's from an HD mouse model, it's supposed to be diseased, then when you look at the traits, it will be fully functional. And the other way around is true. So as long as you have a diseased cortex, then you will drive the dysfunction of the striatum, even though it's from a wild type mouse. So it was very interesting here to see that using these chips and making hybrid networks, which is obviously not feasible using animal studies, we could answer very long-standing questions in the field. There's also this second most recent example I wanted to talk about is, again, this time we switch from neurodegeneration to neurodevelopment. And the question was how the corticostriatal networks develop and how they can keep this very specific organization, this topographical organization within the striatum. So as you probably know, the cortex and projection is very topographically topographical manner with specific sites of projections and non-overlapping zones. And we try to reproduce that in our chips using this specific design where we have two different cortical populations projecting onto a similar striatal chamber here, and the geometry makes it so that cortical axons have their own projection fields, and they are kept within these projection fields, and they won't go into the non-overlapping zone. And what we found actually is that when we use mutant cortical neurons expressing mutant potassium channels that make them hyperexcitable, we completely lose this topography. And now axons, they just spread throughout the striatum, and they will invade any region, and particularly the non-overlapping region. And this is functional. So these axons will go anywhere, and then they will connect new striatal neurons, and they will be functional. So they will activate this zone. And when we make a functional connectivity map, we could see that there is a huge difference between control hyperactive networks that completely redefine how the different populations interact. And that was particularly interesting for us because this is something that is commonly used in the clinics, using mainly functional imaging here, where you say you can drive a connectivity map between the different regions that interact, that are functionally connected. And this is something that we could reproduce in our chip to some extent, of course. And now we are exploring the possibility of making a clinical twin of this kind of experiments using human cells, patient-derived cells. And finally, the last and probably most important project here in the lab is the Mars platform in collaboration with with all of these people, and also academic people, academic university, and also a private company, Flegent, where we are developing what we call a multiplex autonomous assay and rapid screening platform. So it's the same. It's based on the same microchips I was mentioning earlier. So you just again change your design depending on your circuit of interest. Then you have the possibility of doing multiple analyses, again depending on the readouts of interest. We implement stem cells derived from human patients. So this is what I showed before. So we have that, and then now the novelty is to multiplex that. In this example here, we have a chip now containing 12 different chambers with the replication of 12 times the same circuits. And we have connected that to a multi-valve distribution system connected to an area which is made by Flegent. And this allows us to specifically inject a drug of interest in each of the compartments of each of the chips that are on these platforms. So it's either you inject a specific drug in here, let's say in this compartment of this chip, or you can also collect the medium very specifically in this chamber, for instance, to look at biomarkers. So we have a total control of what we inject and collect in each of these chambers, each of this chip, and in parallel. So it allows us now to test drugs of interest that have been first selected using in silico design and screening. So we went from 2 million or so compounds to only 45 of them, defining 45 different families that now we can test in our screening assay. We can not test them just to see if they are active or not. We already know that. What we do here is to test directly if they can correct the different symptoms that we record functionally on our chips. And this led us to the discovery of a new family of anti-degenerative compounds that are called LEAD-TB, and that has led to the creation of the startup company called ATK Therapeutics in collaboration with my colleagues Carsten and Hervé. And now the idea is to use this pipeline of analysis to test and to screen for more, to enlarge the diversity of the family, and to do the functional screening on our on-chip models, so mainly HD models right now, Huntington's disease. And then we'll test for their clinical transferability using the humanized version of these chips, of these HD chips. And then, of course, we will validate all of our results in vivo using animal models on specific symptoms, behavior. In this case, since it's we're talking about Huntington's disease, we are interested in motor impairments, for instance, and tremors. So this is a very important step, the very last step where we need to validate all our finding with the chips. So in conclusion, I think like the main take-home message that I want to, and I want to insist on that here, is that it's possible to create new models for brain disease that are fitting well within the 3 Rs. And because this organ-on-chip system, they nicely bridge the gap between in vitro and in vivo analysis by being more physiological than regular cell cultures, while being more accessible than live brain, live mice, or even acute slices. But what is very important here is that we need to remind that these models remain extremely simplified. They are minimalistic models, and they're intended to screen mechanisms and predict drug efficacy, but they are not truly an alternative to animal research. They help reducing the animal experimentation, but you still need to validate everything that you find in a live organism. So that being said, I want to acknowledge all the people who contributed to this different work. So my current team in Marseille, the past members that contributed, also my former lab in Grenoble, all of the collaborators that I have cited throughout the talk, and the companies, and of course the funding, of course FC3Rs for helping us designing all these projects. And thank you for your attention, and I'm open to questions.
Thank you very much, Maxime, for your presentation. There are some questions, I would assume, right?
Yes, there were some questions. I was wondering, is the company Fluigent? It's a standardized product they are selling. It's not they use it just themselves and do contract research. Or?
No. So it was really a co-development. So very active development from their side, where they provided all of their equipment, knowledge, but also machines and software, so that we could go very fast in designing the platforms. So the machine is currently working as a prototype. It's not let's say commercially grade yet. We still have many problems to fix, but yeah, I'm very positive about that maybe soon it will become commercially available.
Okay, this would be very nice, because there were already some ideas how to use the device for research, like was, for example, the question if plasticity is increased because the cells are close to each other due to the microchannels, and also if, but there was a bit of a question as well, if cells survive well in the device, or if there's a special problem with nutrients or oxygen.
No. So there is no specific problem, not more problems than general problems. And you can perfuse the cells very easily. We are actually trying to do that now. We are even trying to automatize the seeding of iPSCs and the differentiation everything automatized and in parallel. So this is something that is at the beginning. We haven't tried yet, but we believe that it will be possible with very specific. So we'll have to redesign most of the apparatus, but this is something that will not be infeasible to me. But no specific problems.
We have one last question.
Yeah, but was just popping up now. The question was also if it's possible to add glial cells to your device.
Yes, totally. So I didn't present all of it, but actually we have many collaboration now where people are trying to implement different types of cells, and depending on the question, once again, so we have glial cells, of course, oligodendrocytes also, and we're studying also as part of a collaboration the involvement of myelination for the connectivity, because one of the main problem we have here is like pure neuronal culture, so which is not representative of the brain environment. So we are implementing with different cell types one by one to see their involvement and how they will improve the connectivity and survival, maturity, sorry, of these chips. Yes.
Okay, thank you very much, Maxime, again, for sharing your knowledge with us.